asking a question. And now I'm going to go back to my slides and start. This is the start of the lecture for today. So this topic today, recombination in bacteria. This word recombination is meaning something very specific in this context, and it has to do with gene rearrangements or transfers. It's really referring to changing genes in bacteria. And the main reason why we would care about this as medical professionals is that if you have new genes, you have new capabilities. Lobby. Excellent. The first time I did this, I was so fumbly between the different screens and stuff, but I'm getting a lot better now. So if you have new genes, you have new abilities. And for medical bacteria that we care about, that could give them new dangers for us. So new abilities can include things like you resist antibiotics. Go ahead and write that. This could give them the ability to resist antibiotics, to gain new toxins or new dangerous enzymes like coagulase that we saw in lab yesterday. So understanding how bacteria gain these new genes or undergo this recombination process is to protect human health, so that we can understand how these new abilities or these new dangers come up in bacteria. And there are three major ways that they do this. So remember that bacteria normally are going to reproduce asexually. They reproduce asexually. They're doing binary fission. So each daughter cell is going to get the same exact genetic information as the ancestor cell. And that's kind of quick and easy, but they don't get the benefits of sexual reproduction. Brooke, I'm going to put you on the spot. Since I know you've had both gen bios, what is good in a biological sense about sexual reproduction?
What's good about the sexual reproduction or the asexual reproduction?
Yeah, what is the advantage of being a sexually reproducing organism rather than asexually?
I okay, so I think asexual is that it reproduces with itself or that it can be whatever gender it wants, I think.
Yeah, so asexual is there's no other organism required to reproduce with, but that also limits genetic diversity, let's say. Kind of limits your genetic diversity. So sexual reproduction, one of the reasons why it's good for life is that it gives you new genetic diversity or new genetic combinations. So if you have, switch back to pen. If you have more diversity of genes, is generally going to give your population better survivability. Survivability. I'm circling, this is a R right here, survivability. When I write on the iPad, my R's look like V's. For example, this is not diversity, it's diversity. This one is a R. Uh, that's one drawback of me writing on the iPad, just to let you know. So if bacteria can do recombination or have gene sharing, it lets them have some of the benefits of sexual reproduction, even though they normally reproduce asexually. So it's giving them more genetic diversity. It's giving them more genetic options, so that whole populations of bacteria can have better chances to survive. Uh, that's why this recombination process is really helpful to the bacteria. There are three major ways to recombine genes, and these are called conjugation. Let me write them in the order that they are in the slides. Okay, these are called transformation, conjugation, and transduction. Three separate styles of bacterial recombination. One of the things that you will have on Friday's exam is for you to list these, like list the three mechanisms of bacterial recombination, and then that becomes your word bank so that you can answer questions. It's supposed to be a question mark. Answer questions about them. So the A part of the question is going to be list the three styles. And you would write these three words, ideally. And then that becomes your word bank to answer questions about details of these three styles. And that's why I wanted to get straight into this today. So the first method of the first type of way that bacteria can share genes is called transformation. Transformation. And this is a really famous experiment in biology that was headed up by a man named Griffith. So you may have, some of you may have learned about this in high school biology, or possibly Gen Bio 1, called Griffith's experiment. And this experiment, Griffith was using a strain of bacteria that we're going to work with next week that's called Streptococcus pneumoniae. And this is a spelling and a strain that we will talk about more details of it in lab next week. But it's Streptococcus pneumoniae, and you can probably guess from the name that it causes pneumonia. It causes pneumonia in people, and it causes pneumonia also in mice. So there are two kinds of strains or versions of Streptococcus pneumoniae that tend to exist in the world, and one version of it is called smooth, the smooth strain. And smooth strains of Strep pneumoniae have a capsule, that glycocalyx protective layer around them, the capsule variety. And the capsule causes the colonies to appear shiny. That's where the S comes from, is you have shiny, wet-looking colonies. The capsule also, we know now, hides the bacteria from the immune system. Covers up all identifiers on the surface of the bacteria so that our immune system cannot even tell that it's there. So it hides the bacteria from our immune system. So what Griffith started out doing was he knew that if he injected mice, I can draw a syringe here. awesome syringe. Looks just like one. If he injected bacteria of the smooth variety of Streptococcus pneumoniae into mice, it would kill the greatest proportion of them. So we could say that S strain is deadly to mice. These pictures are showing one mouse, but he did it in populations of mice. So he knew this early on. S strain is deadly to mice. There is another version of Streptococcus pneumoniae that's called rough strain. And rough strain colonies look different on a plate. They have a dry, I'm trying to write dry there. They have a dry appearance. Do that over. The colonies look kind of dry and ruffly on the plate, and that's because they're the same as the S strain, except they have no capsule. There's no capsule around them, so they don't have the hydration layer to make them look shiny. They have a dry appearance on a plate. And if Griffith did everything the same with this rough strain, he put the same number of the rough strain into the mice and held everything else constant and did the experiment the exact same way, mice infected with rough strain survive. So we can conclude from this that the R strain of Streptococcus pneumoniae is not deadly. It doesn't mean that it won't make the mouse a little bit sick, but it's not near as dangerous for the mouse's health as the smooth strain. Griffith didn't know why this was. We didn't realize, people didn't know much about the immune system in those days. But they did know that rough strain is not deadly, smooth strain is deadly. What we know now as the reason for this is the immune system of the mouse or of the person can detect and thus protect if the capsule is not there. So if the capsule is not there, our immune system can detect the surface shapes on this bacteria, mount an immune response, and protect us. So having a capsule or not is a big deal to how deadly these bacteria can be. So that's the things that Griffith knew early on during these experiments. He wanted to know what made the S strain deadly. That's what he was really trying to get at. That's what his real question was in the beginning, is he was trying to figure out why is S deadly? And he thought, well, maybe they are making a poison. Maybe they're making a poison.
Dr. You kind of cut it out right there. No, I don't think anyone else could hear you. You kind of cut out because you were moving.
I think I had my hand over the speaker side. Is this better?
Yep.
Yeah, I had my hand over the speakers on this iPad, so I'm gonna try not to do that. So what I was saying is, Griffith knew rough strain was okay, S strain is deadly, but he wanted to know why is S deadly. And he thought, well, maybe they're making a poison. Maybe it's something that's leaking out of the cells that is deadly. So he had the idea that he was going to boil them. He was going to boil the bacteria in the test tube and kill them with heat and then see what happened. So he took a smooth strain with a capsule, boiled it, and this is not an endospore former, so boiling it is enough to kill the strain. And then he injected that dead S strain into the mice, and the mice lived. So what that told him is that whatever is dangerous about the S strain, it has to do with living cells. It's not. It's not probably a poison that's leaking out of them. So he knew that if you kill the S strain and put it in the mouse or the mice will live. So that was some new knowledge. And then he decided to do something different. And I always wonder, why did he do this? Because this is the key part to what he eventually discovered. So he took a dead S strain and a live R strain and mixed them together. So he made a mixture of these two. Mixed them together. And he knew from those earlier experiments that these individually are not deadly. I hear Joseph upstairs. He feels pretty good. He's having a one-man Nerf battle up there. Thumping around. But Griffith knew that individually these are not deadly. He decided to mix them together, put them in the mice, and he discovered that the mice were dying. And this is the kind of result that when you get this result, you think, well, maybe I did that wrong. So he did this multiple times, found that the mice were dying, and then he decided to check their blood. So when he checked the blood of these mice afterward, he was able to isolate living rough and living smooth, which was still a mystery, but it was some knowledge that he had. So he got living rough, he got living smooth back out of the mice. So it seemed to him like the smooth strains, it seemed to him like the rough strains had been in some way transformed into smooth strains. So that's where the name of this mechanism came from, is it seemed like something about this situation was transforming rough into smooth, and it was. Something was. With a lot more experiments, what he found is that I want to erase. What he and eventually some other people figured out was that, well, maybe I can't erase. What he figured out was that in the smooth strain, when you boiled them, they leaked DNA. They were leaking DNA out into the environment. And some pieces of that DNA were able to be used by the R strain. The DNA that was leaking out as these cells were dying was somehow being able to be used by the rough strain. What particular gene would the rough strain need to be transformed into a smooth strain? What gene are they getting?
The capsid gene.
Capsule, yeah. I'm so glad that you said that, actually, because that's going to come up on this test. Capsule is the structure around the bacteria. Capsid is the structure around what?
The virus, right?
Yeah, accidentally hit. Stop. I'm hitting that invite button up there. So capsid is a virus covering structure, and capsule is bacteria covering structure. But all it takes to turn a rough strain into a smooth strain is to get the capsule gene. That's what it took to do the transformation. Griffith did not discover this part himself, actually. But this experiment that he did and the results became famous enough in his day that a lot of other people started trying to figure out, how is this happening? And this experiment is famous for becoming the basis for how bacteria transfer genes, but it also was an early basis for figuring out that DNA is even important in the first place. This was a pretty long time ago. So I want to use, I've put in some blank slides in this presentation because I am sloppy at writing on them. So what I want to do here is give kind of an overview of transformation, how it does and does not act, or what it requires. Let me also say that I usually ask on the exam a question that is asking you about the setup of Griffith's experiment. So you do need to know the setup of the Griffith's experiment and remember like details of rough and smooth and so on. Okay, so for transformation, transformation is actually the ability to move DNA inside a bacterial cell and use it. A lot of bacteria have this ability that they can just take up DNA that's in their environment. Let's make the DNA blue. So here's some DNA that's in the environment, and they could import a little piece of it into the bacterium. And if they're going to really use it, they have to put that little piece into their chromosome. So if the bacterial chromosome is in the red and the new little piece of DNA is in blue, they have to get the piece in, join it with their genome, then they're able to use it. This is the most common way that bacteria can transfer genes. So this does not require the DNA to be related. The DNA can basically be from any source. And we can take advantage of this in experiments actually to make bacteria in some ways do what we want them to do and produce drugs for us, or produce what we want them to produce experimentally. But transformation, the strains don't even have to be related to each other. So the bacterial strains can be unrelated. So the DNA can be from any source. The strains can be unrelated to each other. And you can have a decent length of DNA transferred. There's not too big of a limit on how big this DNA piece can be. It can be a good size piece. Keep in mind, I'm just going to tell this as like a story and not try to write it down. Just because you get a piece of DNA does not mean that it's always going to be useful. I think I'm just going to write useful or not. Okay, green.
I mean, couldn't you potentially give the bacteria like a harmful piece of DNA that would like code for like a bad enzyme that could then potentially like destroy the bacteria?
That's totally possible. It's just more expensive than using a drug.
Okay.
And slower, but that's totally possible. Maybe in the far future we could have genetically engineered people with like well, I'm not going to go there. But anyway, this piece of DNA might be useful or it might not be useful. So I like to compare this to if you came to my house and you got a random piece of paper out of my house that had information on it. It's highly unlikely that that would be useful to you. Like if you came in my house right now and you grabbed the first piece of paper when you came in the door, it would be a Taco Bell wrapper. Or the next one you came to would be coupons that go in your mailbox that you didn't ask for, or a save the date card for a student wedding that's already over. So out of all the million, probably not a million, all the paper in this house, if you're comparing DNA to papers in the house, the bacteria can't tell whether this is good or not, but they can just pick it up and try to use it. So maybe if you were super lucky, you might come in my house and find my birth certificate or something like that, or my social security card, or if I was dumb enough to print out the email that that I send to Han every few years with like, if I die, these are all the passwords. That would be the best thing that you could find. But it's unlikely that it's useful. But there's a chance that it's useful. And if it is useful, that's a huge advantage to this bacterium. So in the rough strain and the smooth strain, the gene that was picked up for the capsule, sometimes that will prevent the death of the bacteria. Not always, but sometimes it would prevent the death of the bacteria. And then if that's true, those bacteria live and they pass on the genes to their descendants, and now they're more dangerous to people, if that makes sense. So there's no selection of the bacteria like, oh, I want a capsule gene. There just happened to be transfer of a capsule gene. And most of the DNA that would be picked up in this way or any of the other ways is going to be a dead end. But if you get that one magic lucky gene, that means the difference between life and death. Okay, so that's transformation. I don't spend as long on conjugation and transduction because the experiments to show those were not as famous and not as awesome to me as the experiments to work out how conjugation happens. But conjugation is the second method, and conjugation requires the bacteria to have a pilus built between them. So remember that a pilus is that hollow connecting tube that allows bacteria to send things from one cell to the other cell. So this requires a pilus. And what goes through the pilus has to be a plasmid. It has to be a particular type of plasmid. So this allows transfer of a plasmid DNA through a pilus yeah, I guess we'll stick with related, to a related bacterium. And they basically have to be in the same genus to do this. This is in the whole world of bacterial recombination, conjugation is less common than transformation because it has a lot more requirements. So these two bacteria have to be basically the same kind of thing, like E. coli and E. coli. And it was first discovered in E. coli too. So they have to be related to each other. They have to be able to make a pilus. And the only DNA that can go through the pilus is the plasmid.
How does the pilus get created? That just kind of just like pop up, or like how's that like form
No,
separated?
so it's kind of expensive to build a pilus, and bacteria don't just build them randomly. They have to kind of sense the presence of a nearby cell.
Okay.
So sometimes it's the presence of another cell like it, or if the pilus is being built to a host cell or a cell that's going to get infected, there are ways that the bacteria can sense the presence of its victim, let's say.
Okay.
Yeah, that's some of the labs that I used to work with in graduate school were working on that kind of question, like why do they build the pilus in the first place? But it's always on purpose. Like you don't want to build a bridge to nowhere.
So like then is the pilus made out of like the cytoplasmic membrane? Because in the picture it's just their cell walls are now
Yeah,
tube.
it's actually like making there's a whole structure here that our book doesn't show. That's like you build the base of it, and then it's a hollow tube, and you send the parts out through the tube, and then they assemble on the other side, and it ends up connecting the cytoplasms.
Okay.
It's really cool. I talk about it a little bit more in the majors class, but it's a complicated process to get it to build, but it's beneficial to have the ability overall, because once in a while you can get something useful out of it.
And then once the pilus is built, is it can you easily detach from it, or is it kind of like a permanent joining of bacteria?
It's not permanent. I don't know if anyone is studying the taking down of the pilus, if that makes sense.
Mm-hmm.
Not aware of that. But you can probably tell, like, I really like the idea of sending weapons through a pilus. That like totally captured my imagination when I was in graduate school. But it seems like this system to send genes, this is the original purpose for a pilus. And then later it became useful to also send weapons to a victim cell. So conjugation is a way that bacteria that have a pilus, or they have pilus ability, let's say, can send a plasmid to another bacterium like them. Something else that is different about this than transformation is you kind of have a limited length, because some parts of this plasmid are other genes that are like plasmid maintaining genes, and you don't have an unlimited length ability like you do with transformation. But that's the basic info about conjugation. Conjugation is more, it's more of a gram-negative thing. It's more common in gram-negatives than gram-positives, because gram-positives don't make pili, as far as I know. So the third, this blank page was for me to write about conjugation, but I already did write what I want to write about conjugation. So we're going to skip over this blankness. Oops. So the third way that genes can move between bacteria is via transduction. And be really careful when you're writing or answering questions about these methods that you don't mix up transduction and transformation. Those are two different things. And unfortunately, we also have on this exam transcription and translation. And all of these four are similar to words, but they are not.
Oh, I think your thumbs over the speaker thing again. Could you repeat the last like sentence, please?
Yeah. I don't know what happened actually. I did not have my hand that close, but what I was saying is transduction is the third way that I'm going to talk about next. It's not the same as transformation. Students tend to mix those up sometimes when they're writing about them. But also be careful on this exam that we've also talked some about transcription and translation, and none of these four words are the same. They all mean something different. So be careful if you're writing any of these trans words that you've got the right word that you mean on the exam. So transduction, what the first thing that you should remember about this is that transduction requires viruses. It's viruses that are moving DNA between bacterial cells. Viruses are moving the DNA between related cells. And the specific viruses that are doing this are these bacteriophages that we talked a little bit about during the virus section. So it's a normal process for a bacterium like this E. coli here to get infected by a bacteriophage, a virus, and the virus injects its genome. That's the penetration step. And the virus genome will be inside the bacterial cell along with host DNA. So we've got virus DNA is in the red, and the host DNA is in this pinky color. So when the viruses are self-assembling, when we have viral self-assembly, viruses are really dumb. I know they don't have brains, and bacteria don't have brains either, but viral self-assembly can be really sloppy. So what happens when these viral parts are coming together in the cell is that you get the head capsid going together with the genome first. And the way that this works is you measure the virus, essentially measures a certain length of DNA that's going to fit into that head capsid. So they are packing up a certain length of DNA. They cannot detect where the DNA came from. It's just a certain length of DNA. So this would be like if I asked one of my kids to pack for a trip. I said, get your little travel bag, which is a free King duffel bag that we got like at a game or something. Get your travel bag and pack for the trip and put clothes. It would be like if they just go to a random drawer and like get enough underwear to fill up the bag and then they say that they're ready to go, like that a tiny kid would do. Viruses are kind of like that when they pack up to go, like, yeah, I got enough DNA, I'm ready. And this one over here, get red. This one can't tell that it accidentally got a piece of the host DNA. So, yeah, I got DNA, I'm ready to go. That's the wrong DNA. So I'm going to the next slide now. It's just a continuation of this process to show what happens next. So here come the viruses. Like here come the 3 on kids out of the house. They got their suitcases. I'm ready. And one of them has totally inappropriate material packed for the trip. And that virus, it can still attach to a host cell and it can still do penetration and send the genome in. But what is happening is that this host cell is getting bacterial DNA. This host cell is getting bacterial DNA, not viral. So I like to think that this cell is doubly lucky. Number one, it didn't get infected by a real virus, so it's going to live, and that's already great. But that DNA that it got even has a slight chance of being helpful. Oops. There's a slight chance that that's a helpful DNA. It's getting something. It's like someone brought a paper to you out of my house, and it's probably garbage. But it could be something useful. So if that piece of DNA gets integrated with the host chromosome, it's usable, and it's possible that it could be helpful. But with the number of viruses that infect bacteria on a daily basis, having this be useful, it's rare, but it happens. And it's one of the three major ways that bacteria can get new genes. This is transduction. So I want to, yeah.
What's the main difference between this and then the lysogenic cycle? Because the bacteria or the bacteriophage is both injecting its DNA into the bacteria.
Yeah, so the difference between this and lysogenic cycle is really only that the piece of DNA is not viral.
Okay.
That's really the only difference. And lysogenic cycle, like the discovery of lysogenic cycle basically goes along with the discovery of transduction. And that was kind of an early on discovery, and the realization that that could happen in people with human viruses didn't come until a lot later.
Okay.
But the general concept of it was worked out in E. coli, I feel like back in the 50s or 60s or something like that, pretty early on. So overall, our takeaways from transduction, what's required for transduction? You have to have a virus, and the virus is going to move a defined short length of DNA, short length of DNA between, that means between bacteria of the same genus. They have to be related. So there's a lot of requirements here. You've got to have a virus. You've got to have, there's limits on how big of a piece of DNA could be moved. And it's got to go between bacteria of the same genus. But it's one of the three major ways. I feel like I moved on from that slide too quickly. The reason I'm not going back to it is when I'm, actually this is a Teams recording, so I can go back. If I do explain everything recording, I can't go back. So make sure you had time to write that down.
What would happen so then if the bacteria are not of the same genus, so then would that bacterial DNA of like the streptococcus, would that just kind of be lost in like the staphylococcus? Just for like what would happen with that?
So, which are you talking about? Transduction?
Yeah, transduction. Like if the
Yeah.
bacteria are not of the same genus, then it's just like another different infection.
Yeah, if the bacteria are not of the same genus, um, like viruses are specific to different hosts. So this virus is specific to infect E. coli, but
Okay.
it could not attach to Staphylococcus.
Okay.
Yeah, that's why they have to be the same genus or related, because they have to be both infectable by the same virus.
Okay. Okay. Yeah.
Yeah, that's a good question though. So now that I realize I can go back in this recording, I want to go back to kind of compare and contrast with conjugation. So conjugation, you've got to have the same related type of cell. Um, they have to be able to make a pilus between each other. This one, it's a plasmid that moves, and the gene that is involved has to already be in a plasmid for it to move to the other cell. So that's a little bit different than transduction. And if we compare back to transformation, this one is quite different style because the DNA is just coming from outside the cell in the environment. So I'm going to add in this word. The DNA can be in the environment, just kind of lying around. And someone did a study of hospital drainage and sewer systems, and there is a lot of DNA of microbes in that environment, and the water drainage and sewer systems of hospitals are one of the major places in the world where this happens, because you've got a lot of live bacteria, a lot of DNA from dead bacteria, and that's a kind of a scary situation.
Mm-hmm.
Uh, okay. This slide is from your book, and it is technically listing and comparing these three ways. I do not really love how they describe it because I feel like the language is unnecessarily complicated for this style of class. So if you're looking at factors involved, like that is the same stuff that I have basically told you, except they're using language that does not match the general language of this book. So I'm not going to cross that out because it is helpful. I am going to cross this out. Don't worry about direct, indirect. But it's this part over here that I just kind of wanted to talk through a little bit. What kind of genes are being moved? So drug resistance, toxins. I don't need you to match these with the style. I just kind of want you to know these are some of the beneficial lucky genes that bacteria could share with each other through these methods. And being drug resistant is dangerous to people. Toxin production is dangerous to people. Getting a capsule that can hide you from the immune system is dangerous to people. So this kind of research about how this happens directly relates to human health because we would like to prevent this. So if you can prevent how fast this happens, in the end, that's going to protect human health. So we want to prevent, and then in a more basic way, understanding how it happens um, just gives us greater scientific knowledge about bacteria generally and how we can use them or how we can stop them. So what I want to do here, we've got about 10 minutes. Do you guys have questions about the exam Friday or things that you want me to go over?
So for for the required things that you said to study, so you mentioned ATP production and like the fermentation, aerobic and anaerobic cycles, but you didn't mention anything about the NADH plus and FADH2. Do we also need to know how many of those molecules are produced as well?
I
Or do we
don't
really
think
care about?
so.
So okay, we just really care about the ATP.
I'm pretty sure for this class, it's more of ATP. Although NADH and FADH2, that info might be in multiple choice on one question or something.
Okay.
But the ATP is the more important factor for this class. Let me maybe see if I can. No, we don't have a printed out. We don't have a printed out exam sheet. It was just what we wrote on the board. Let me get it on my phone. Took a picture of it. While I'm doing that, my plan for Friday is I will be there in person to give you the exam, but I'm going to probably have on a N95 style kind of mask, and I will want you all to sit really far from me. So those of you who sit in the front row, if you would go sit in the back row again for the exam, that's just to be really cautious while we're waiting on the results of Joseph's test. What was I wanting to do right here? Oh yeah, look up the exam topics. Okay, I'm gonna turn my camera back on for the time being. Yeah, now you can see me again. Other questions you have about the exam? There, that's where the camera is. If you, I'm going to stay on a few minutes in case you need to talk to me or you think of a question. But if you think of other questions, just email me today or tomorrow or Friday morning if you must. But we'll have the exam on Friday at the usual time, usual way. And I am going to post this recording into the page for today in Canvas so that you can go back and refer to it if you need to. Also, if we ever did have to go online, this is what I would do, like daily for our lecture. This is what I would do because I think I've kind of got this working well, and that's what I decided on. But we're not that close to going online anyway. So if you don't need to ask me anything right now, I will see you Friday in a annoying mask and far away from you. But if you do want to ask me anything right now, just stay on and ask. And I'll see you all Friday.
Yeah. Have a great rest of your day, Dr. Ong.
Yeah, should be good.
Dr. Ong, this is Brooke.
Uh-huh.
I have a few questions about the exam.
Uh-huh.
So when we are talking about the aerobic, anaerobic, and fermentation, do you want us to know also like the waste products that come off of each and every single one of them? Because I know for the electron transport, when when it is anaerobic, it uses electron acceptors to
Uh-huh.
produce the waste product. Well, to produce ATP.
Mm-hmm.
I guess my question is, I don't know. Never mind. Forget it.
Well, that's a good question, because I think I was a little bit unclear about that at the end of lecture Monday. So for anaerobic respiration, the only waste product that you need to know is CO2. Because what the specific waste products are for anaerobic respiration depends on what the electron acceptor is. So all the styles of glucose metabolism will make CO2 as a waste product. For aerobic respiration, it's CO2 and water are the major wastes. For fermentation,
Mm-hmm.
for fermentation, the major waste products for fermentation generally are CO2 and acids and alcohols.
Okay, so you don't want us to know like the NO substrate 3
No,
negative.
that's a little too detailed for this exam. There are certain of those, like the sulfur waste products, matter when we get to gram negatives.
Okay.
When we're studying the gram negatives in the lab in 2 weeks, we do a bunch of tests that detect sulfur waste products. But when I need you to know that, that will be in lab and not on this lecture exam.
Okay.
Good question. Chelsea, did you see anything else in the AMP2 video this morning that was not great or messed up?
No, I thought it went really good after I got those PowerPoint.
Okay, good. The reason I don't do that for this class is I don't have any videos already for this class. So this style is probably better, but I had to literally go to the doctor during class with Joseph, so I was hoping it went okay. Well, I'm going to sign off. If you all have other questions, just let me know by email. And I'm glad this worked okay today. So I will see you soon. Bye.